The genetics of complex traits is the real issue of genetics. Let's say the genetics of Mendelian traits is interesting, but does not account for most of the interesting things we want to deal in humans, and we have to understand not only the cases for which we have nice models, but the cases that we do not even understand properly. So here today we are going to see how it works, the genetic of complex traits, how we know that a given trait is transmitted. There is a heritability, meaning that it runs in family because of genes. How today lots of geneticists are working on that, and how this kind of work is going to be done in the future. When we try to understand genetic disease, we have to see that in many cases there are complex diseases that do not follow the rules of Mendel's law, that do not follow single ways of inheritance. We can think on many cases of diseases or traits or whatever, and we see that they run in families, they have to be in a certain way inherited, but they do not follow a single model of inheritance that would follow diseases and that would follow the molecular view of our genome. In general, complex diseases, and this is the most important thing, they have high prevalence. They affect lots of people. And here we have cases of arthritis, we have lots of different autoimmune disease, all the psychiatric diseases, autism, schizophrenia, bipolar disorders, cancer. There is a genetic base in cancer, and in most cases there is also a complex susceptibility to it. We have the cardiomyopathies, meaning that we have lots of cases, diabetes for example, in which it's extremely interesting that they have a relevant genetic component, no simple model of inheritance, and we have in all the cases several genes with small contributions. We are not going to talk on the gene for, as we did for example for cystic fibrosis, but genes of susceptibility to schizophrenia for example. There are multiple alleles interacting among them, interacting with the environment, and these complex interactions are at the base of the genetic of complex diseases. To begin with that, we need to recognize to which extent a given trait is inherited or not. And for that, we have to work out what we call the heritability, which is the proportion of a phenotype variance or the variation of a given trait that is accounted by the genome, by the differences in the genome. Is the degree of variation in a phenotype trait in a population that is due to genetic variation between individuals in that population. In general, it is written as heritability small h square equal to the variance due to the genes. A is for additive effect of several genes, divided by the total. In this case, if we have an h square equal 1, means that high heritability indicates that the environment doesn't have a strong importance or very little importance in the phenotype variation, and h equals 0 is driven basically by the environment. In this case, the genome doesn't account for the variation of the trait. to work out, to calculate the heritability of a trait is to give an idea to which extent this trait comes from genes, comes from the environment. We don't have to have a strict idea if we have a heritability of 0.5 saying 50%. No. Means this value depends on how it is calculated, in the population, in the genetic background. So it's a general idea of the relative importance of genetic factors in relation to environmental factors. In many cases, this is being studied through comparing monozygotic and dizygotic twins. When we compare them, what we take into consideration is that the monozygotic share genes and environment, and the dizygotic twins share one half of the genome, but share the environment in the same way as the monozygotic. And this allows us to calculate through the difference of the resemblance of ones in relation to the other, to calculate the heritability. In this way, heritability will be from 0 to 1 depending on the trait in general, and also remember on the way it is calculated in the population that is calculated. These are a general view of cases in which we have to the left the cases of very strong genetic base, so these are the Mendelian, like Duchenne muscular dystrophy or cystic fibrosis, and if we go to the right, every time is more and more importance of the environment. So this line is a rough idea of the values of age, and here what we have are different conditions in which we have varying degrees of importance of genes versus environment. It is interesting to see that there is strong heritabilities for some human traits, and this is interesting because this means that we have to work to deal, to dissect, to recognize which is exactly the genetic base of that. If we have for a given disease, diabetes, let's say, a given heritability, then we have to go and do studies to dissect this genetic of the diabetes. It is interesting to note that there are many studies in which people have been trying to understand the existence of heritability for physical traits, for diseases, for all kinds of conditions, but also for psychiatric diseases for which still now we do not have a deep knowledge of the genetic basis of it. For example, there are psychiatric conditions for which we know that we have a heritability, let's say, of 0.7, which is quite high, but for which we do not know yet which are the specific genes at the base of that. But something there must exist. And here you see also for psychological conditions, meaning that we have lots of interesting issues in which the genome has to play an important part, and being that complex phenotypes, in most of the cases, we are not able yet to work out in a proper manner the relationship between the genotype and the phenotype, and then to recognize which are the genetic variants at the base of this specific heritability. In the last years, there has been a lot of work in what is called genome-wide association studies, or GWAS. In these studies, the idea is very simple: to gather a population of patients, a very similar populations that are of normal individuals that may be used as control of this population, and then to look at the genome of both populations and see where in the genome there are discrepancies that could be related to the presence of the disease. The idea then is to gather lots of information on lots of patients, looking at really deep at the genome, and then to do this analysis. In general, so we have the two populations, the genome analysis on them, and then statistical analysis. In a simple case, we can see that in one of the two populations, in the control or in the disease populations, we have a higher frequency of a given variant in the genome. What means is that this variant could be at the base of causation of this different susceptibility. And there are lots of statistical ways of dealing with this data. Imagine that in all the cases it's huge amount of data, and in this association analysis we have many different diseases and traits. We have a very large number of individuals and looking at deep in the genome. Then we have lots of statistics, and at the very end we end up with a plot in which we can see the whole genome, and in some places we see that there are peaks in which we see something unexpected, in the sense that the frequency of these variants are much higher in one of the populations, let's say in the populations of patients, meaning that it is very likely that in these portions of the genome there will be genes of susceptibility. In general, with that, we are able to work out the cases in which diseases with high frequency are being dissected with variants in the genome that explain the susceptibility they may have. Here in this plot you can see some of the examples in the diagonal, going from the top left to the bottom right, and in this case you can see from the clear monogenic to the very complex cases in which the variants that are really at high frequency may account for diseases that are also at high frequency. The effect of these variants vary, and the ideal is to find the variants that have a deeper, a very strong effect, but these are the ones that are being much easily found in all the studies. And right now people working in the genetic of complex diseases are trying to find the ones that are already in the lower part, meaning in the susceptibility not being very, very strong. To have information on that, there is the GWAS catalog, in which you can see to thousands of cases in which they have been worked out. And today we already have maps of genetic diseases, maps of the genome with the susceptibility genes, and the genetic of complex traits is a field in which people still are trying to discover new places in the genome, and what is more important, to understand these places in the genome, how they are related to the disease. Because if these regions exist, means that the genes in these regions, they have to have some connection to the physiology of the specific disease. To which extent the genetic of complex traits is properly solved today? We have to recognize that only partially, because we have many cases in which we have the susceptibility, we have several genes, in some cases quite a lot of genes related to that, but in many cases we still are not able to explain properly the heritability that we know exists. For example, we have cases, maybe the best known is the genetic of diabetes mellitus type 2, in which there is a list of genes. Some of them are very interesting because people didn't know these genes were related to the etiology of the disease, but now we know they are. Meaning that every time in the future, what we have to do is to keep on developing this kind of studies, but there is a need to understand much better the physiology of the disease and how the genes that people are discovering are related physiologically to the disease. So this is a field ongoing. There are still an important part of the genetic basis of complex characteristics is still to be understood. Today we have with us Anna Di Rienzo, professor at the Department of Genetics at the University of Chicago. Her lab focuses on the genetic architecture of traits and diseases that vary across populations and tries to understand natural selection and how natural selection is related to the adaptation. To this end, they use empirical population genetic and functional genomic approaches with computational analysis of genome scale. They are doing a lot of work trying to understand the patterns of human genome variation and to understand also disease mapping. In this interview, we are going to focus on the genetics of complex traits. This is a very hot topic in genetics and in biology, with lots of efforts trying to elucidate the genetic architecture of complex traits, including, of course, complex diseases. At the very end, what is attempted is to understand how the genome shapes the phenotype. Anna, genetics is provided lots of new results in the dissection of complex traits, like diabetes, obesity, psychiatric disease, by doing what is called a genome-wide association analysis. Is this strike fruitful? Is it going to solve the problems related to the understanding of complex traits or the relationship between genotype and phenotype?
Well, the genome-wide association studies are exactly about trying to connect genotypes with phenotypes, and specifically the disease phenotypes that you have mentioned. There are a number of very interesting results that have been shown in some diseases like type 2 diabetes, the diabetes of the adult age. There is one gene that is known to be very important, and lots of genes that have small contribution, that have a small influence on the probability of developing the disease. In other cases, the situation is much more complex. There is an even larger number of genes that can influence your risk, your probability to developing the disease. Now, one challenge that has emerged already is that for many diseases, the number of results that you find in terms of the number of genes that you find to be associated with the disease can be rather small. And they explain, together can change your probability of developing a disease by a very small amount. So from a practical standpoint, that means that we are still far from being able to use genetics to make accurate predictions of disease risk, and therefore to make predictions that can change the clinical practice, what can happen in the clinic. But at the same time, there is certainly an increased understanding and knowledge of the kind of biological pathways, the groups of genes, the metabolic pathways that are important for some disease compared to others. This is clearly very different across the diseases. I think that so far, many autoimmune diseases, those diseases in which the body produces antibodies against some component of our own body, have made a lot more progress in the sense that we understand a lot better the genetics of these diseases. For metabolic diseases, perhaps psychiatric diseases, asthma, there are many complications that are still not completely accounted for.
Why is so complex? Why are we not much better in making predictions just looking at the genes?
Well, with regard to the ability of making a prediction, the main problem is that the disease risk is the result of lots and lots of different genes, each making a very small contribution, meaning that each gene changes your risk by a very small amount. And of all these genes that are underlying a particular disease, we know only of a small minority of them. So that's a first challenge in terms of taking the genetic data and using it in practice in a clinical setting. There is also another problem, and that's certainly the case for many diseases, for example, psychiatric or behavioral diseases, and that is that the environment, and by environment I mean the physical environment, but perhaps more importantly, the social and cultural environment and the economic environment may have a major contribution, much bigger than the disease itself, than the genetics itself. For example, let's think about type 2 diabetes. Type 2 diabetes is a disease that if you are diagnosed early enough, if you change your lifestyle and diet, you can actually reverse the disease. So, for example, if you're diagnosed with type 2 diabetes, if you start exercising very regularly and a lot, and you change your diet to have a lower calorie diet, you can actually reverse the disease. So your genotype, your genes do not change, but if you change the environment, your environment, you can change the outcome. And this is very important to remember always, right? That the environment plays a very important role. Another way in which the environment can play a role and can complicate things is in what we refer to as gene by environment interactions. And this is probably very important in diseases like asthma. And what does that mean? Well, it means that you may have a certain genetic variant that increases your probability of developing the disease only if you are exposed in a particular environment, but not if you're not. Now, why is that a challenge? Because it's very difficult to detect those gene-environment interactions. It's very difficult statistically to be able to distinguish a variant that affects the disease risk only in some conditions. And so some people think that that's part of the reason why, for example, we still understand comparatively little about the genetics of asthma, even though many genes have already been associated. Yet another possibility is that instead of having lots and lots of genes, each with variants that are quite common in the populations, and changing your disease risk by a little bit, that there are variants that are very rare in the population that can affect your risk to a great deal. And that is something that a number of people have been working on. And there is a lot of interest in that area. It has been applied very extensively to a number of psychiatric diseases and other diseases. And it's clear that this model is important to a different extent for different diseases. So the complexity comes from the fact that the etiology, the causes of the disease are complex. They are both genetic and environmental, so there is this complexity. And in addition, for both genetics and the environment, there is a diversity of possibilities, of possible factors that can increase your risk.
And you see the research that is being done now in the right track to solve these complex problems, because you are showing us as a very, very, very complex issue.
Yes, it is a very complex issue. I can't say that we know what the right direction is. I think that what many investigators are hoping is that whether or not we fully understand the genetics of the disease, that we might have enough of an insight to the genetic basis of the disease so that we can develop drugs that target that particular aspect of the biology of the disease that can serve as a treatment.
No doubt. If this research is so complex for cases like type 2 diabetes, it has to be much harder to get into psychiatric or psychological issues.
Yeah.
Is it going to be fruitful? Is it going to be really worth the genetic analysis to try to understand the genetic basis of behavior?
Yeah. So this is really a difficult question, and I'm sure that there is no one objective answer. We all have different opinions. I think what is going to be necessary for psychiatric diseases and behavioral traits is a better understanding of the biology of our brain and our nervous system and how it is shaped by the environment in which we live. And I think that there is a lot of progress that is being made in that area, so that when we do a genome-wide association study for a psychiatric disease, we have better ways to interpret the results, and in particular to tell apart the true signal that has a biological meaning from signal that is probably spurious. So I think that there is a lot of work going on in this area. It's very challenging because not only these conditions have a very significant environmental, especially social, economic component, but also learning about the biology of these diseases is complicated because you need to have access to tissues that are rarely available. So that's an additional layer of complexity that needs to be considered.
Okay, so we have seen the complexity of trying to solve how genes are at the base of ourselves. Genes, and as Anna Dirienzo told us, with the interaction with the environment and with very complex setting that genetics, biology is trying to solve, but we have to understand we are still in its infancy. Thank you very much.
You're welcome. Thank you.
No doubt the genetic of complex traits is one of the most challenging points in biology and in genetics today, because it has a lot of implications not only in its understanding but also in the applications. Today we have with us Greg Gibson. He's professor and director of the Center for Integrative Genomics in the School of Medicine in Emory University in Atlanta in USA. He was trained as a geneticist in university, and then he worked in Basel and then also in Stanford, and it was professor at the School of Biology at Georgia Tech since 2009. His research focuses on broad interest areas on quantitative genetics, quantitative evolutionary genetics, the understanding of susceptibility using also not only DNA sequences but transcriptomics, and trying to understand how these expression profiles may be related to genomics, but with a very clear applied interest in health. In this interview, we are going to focus on complex trait, but mainly on its importance, what is today called the personalized medicine or personalized genomic medicine, both in what is called precision medicine and what Greg has coined as predictive health, meaning that using the knowledge we have on complex traits not only to understand the genetic of complex traits but to be used for improving our health. Greg, genetics is providing lots of new results in the dissection of complex traits, diseases like diabetes, obesity, psychiatric disorders, and what is used in general is what is called the GWAS or genome-wide association studies. Is this track fruitful? Are we learning things? It is worth going in this way?
Well, first of all, thank you very much for inviting me here today in this beautiful setting. It's fabulous to be here. You know, I'm a really big fan of GWAS. I think it's really revolutionized, and I use that word not too carefully. It's revolutionized our understanding of genetics. I think I was lucky enough when I was a student in Basel 30 years ago to be part of the revolution that understood how genes regulate development. And now, 25 years later, we've had a new revolution about understanding how quantitative genetics works, how genes throughout the genome regulate disease and evolution and so much about who we are. You know, Peter Vischer is one of the leaders of the field. He wrote a review with Mark McCarthy 5 years ago after 5 years, and they said for the cost of about an aircraft carrier, so about $250 million, we have found thousands of genes influencing a wide range of complex traits, and argued that that was an enormously productive investment, and I can't agree more. Compared to the way we did it 30 years ago, 40 years ago, it's so much more powerful, so much more illuminating. I think the most important thing it's done is that it's really distinguished between three models that existed for the genetics of complex traits. So one was common disease, common variants, was the idea that every disease, diabetes, cancer, whatever it was, had about 20 to 50 genes that were important, and each one of them would explain maybe 5% of the variation. It turns out that's just not the case. So the two alternatives left are something called rare diseases of large effect, rare alleles of large effect. So if you came from psychiatric genetics or early disease, early onset congenital disorders, people would say that, look, every different case is a different case with a different mutation in a gene, and if without that mutation you won't have the disease. The other viewpoint is what's called the infinitesimal model, which is that there are in fact thousands of genes influencing every trait, and they have very, very small effects. Maybe an effect as small as 1/10 of a millimeter of height, or an extra day of staying in school. And now I think it's absolutely clear that the infinitesimal model is winning out across all of the different classes of disease and complex traits. And that's a very profound understanding, because it tells us that genetics is so much more complicated than we ever thought before.
But this complication, it seems that it won't be able to have an application on that, because if we have thousands of genes, at the end we said, well, that's too complicated to have any possibility of using this information for health or medicine.
Yeah, so it's a bit of a cup half full, cup half empty situation. But I think the more we know about the core genes, the more we know about ways to approach complex traits. So there is a new paper that came out about 3 months ago, so in June, I think, of 2017, from Jonathan Pritchard in Cell, talking about something called Omnigenics. And what he points out now is we know that there are about 2 or 3% of all of the SNP variations in the genome probably influence any given trait. So that's 100,000 for height, 100,000 for obesity, 100,000 for diabetes risk, 100,000 for educational attainment. It's an enormous amount of variation, and any single variant explaining a tiny fraction isn't useful. But he also argues that the reason it's that way is because there's a core set of genes, maybe 100 or 200, that are operating in a particular set of cells that are critical for that disease. And if that gene's involved in, say, signal transduction from the immunoglobulin receptor, then clearly it's critical for the biology. But anything else in that cell will ultimately impact upon that level of activity, and that's the background, the very, very small amount. So that background gives us the modifiers. It gives us a sort of a standard against which all of the more highly penetrant and environmental effects operate. So you can be protected because of your background, or you can be at high risk because of your background, and then it takes something else major to really push you over an edge into disease, is this new idea. And sorry, carry on.
But you don't think that being things so complicated will make the intervention much more difficult?
You know, I think it's also important to realize that the intervention doesn't even have to target variable things. Right? So the orangutans behind us all have 5 fingers. You're not an orangutan without 5 of these long fingers doing what they can. But of course, that's what makes them who they are, without variability. It's the same in disease. A lot of processes don't have to be variable. One reason that I do more than just genetics is because it gives us an insight into things that aren't captured by variation. So in Crohn's disease, for example, which is one of the diseases I study, we've learned from transcriptomics that the extracellular matrix and the mitochondrial function are absolutely critical. But if you do GWAS, they don't get hits. They're not implicated. Okay? So it's a whole integrated system that's changing in any of these diseases. And GWAS is just a light on that. But still, the pathology is very specific. What's going on is much bigger than just the individual variants. So in the end, in the cells and the cell biology, it's just as targetable and analyzable, and we can make interventions despite the complexity of the underlying genetics.
That's the point you have introduced. It's a very interesting point in using, you have been pioneer on that, using transcriptomics. That's the genes that are really working in a given cell, in a given tissue. This information is independent of the information in the genome. So the information that the transcriptomic profile and the genes, the GWAS, are independent?
I think independence is too strong a word. There are 4 reasons, I guess 3 or 4 reasons why I like to study the transcriptome and not just the genome. So one is the one I just gave you, that actually it's more than the variable things that matter. So another one is we want to open up that black box between the genotype and the phenotype. And if the genotype, we want to know what the genotypes are doing at a molecular level. Okay? So if you want to understand why a Ferrari is different from a Ford, just having the parts list isn't going to tell you. You have to understand how those different parts of the Ferrari make it faster and stronger. Okay? And so that takes studying downstream. So we can do that with transcriptomics. Another reason is because it turns out that about the majority, maybe as many as 90% of these GWAS variants are regulating gene expression. So it makes sense then to study how they're regulating gene expression, to study the gene expression absolutely directly. And we can do that with the same sorts of tools now. We can study all 20,000 genes at once and see what they're doing in a very coordinated manner, and it gives us new insights. And I guess the other reason is I just think it's more fun and more exciting, actually, to study the transcriptome than the genome.
The example you have put, I remember the classical book of human genetics by Vogel and Motulsky. They put that example of the cars. To tell the difference between cars is extremely difficult.
Yeah.
To tell the difference between an operating and a non-operating car is much easier. Yes. And this is the interesting point. The genome, the transcriptome, and we have to talk on another point that you have also been working. It's the environment, what now it seems that we should call exposome. Yeah. I don't know whether the word is widely accepted or not. The point is, the exposome no doubt is important for health. Why we are working so much in genetics and maybe there is no much effort on the exposures we are?
Technology is because we can, I guess, is the answer. I mean, this genetics, this revolution has happened because of a conjunction of computational power, statistical know-how, and just flat-out technology that can let us measure 500,000 genotypes for 50 euros per person. So we're not there with the proteome for sure, and the metabolome, which is really what's critical for the exposures. Again, that technology is great for looking at about 200 things, but doing it genome-wide to look at 50,000 metabolites is not possible yet. But I think it's a little bit more fundamental than that too. It's a little bit theoretical. So as geneticists, we partition or we divide the sources of variation into genetic and environmental, and we allow for interactions between them. But the genetic we can study now, and it's fabulous. And we will soon have millions of sequenced genomes. What's the equivalent on the environment side? Okay. Well, let's think about partitioning an environment. You've got things you can measure, like how warm it is, how much daylight there is, what elevation you're at, what your socioeconomic status is. Okay, they're very broad measures. And then there's a component that's stochastic. How do we measure that? How do we measure whether you had a trauma when you were 3 years old? How do we measure whether you've been exposed to some toxin that you got through sitting in a rainstorm with from a cement factory? We don't know how to measure those things or what to measure yet. So right now I think environmental exposome stuff is done one component at a time, and it's expensive. The beauty of genetics is it's everything and comprehensive. Of course, if we could study that in the exposome, we would do it, but hopefully we'll get there. But I think that's the reason why. It's not because it's not important, it's critical, but we can't do it.
A close friend of mine used to pose in the following terms: the genome is huge, but it's finite.
Yep.
The environment, we don't know how big it is.
Yep.
At the very end, precision medicine, how all that is going to be used into the health.
Yeah.
How can we put that? And the promises that genomics has been doing into health, are they going to be fulfilled?
So actually the reason I'm here in Barcelona these 3 months is to think about these issues. I think there's a lot of hype around personalized medicine, that you're going to be able to say to somebody, you will die on your 73rd birthday of kidney failure. That's garbage. That's not going to happen. But what we can do is, I think, 2 areas that I'm really intrigued by. So one is risk classification. So we can say, look, you've got above average risk of cardiovascular disease or above average risk of depression. Now what do you do with that? So one thing you can imagine is saying, okay, if your blood pressure is extremely high, you have high hypertension, and yet your genetics says that you shouldn't, then maybe that's a pointer that says, look, there's something in your lifestyle that's addressable. Or the other way around, maybe you say, look, my genetics of type 2 diabetes says that I'm at very high risk. Metabolically, I'm absolutely fine right now, but then you'll be more vigilant about that. So I think that's a level that we could engage people. Not everybody is going to become engaged. So some people will respond to that, others won't. But I think that that's potentially useful. Or even something more pragmatic, such as a variant that says I've got a high risk of deep vein thrombosis, might change how you fly and things. So that's usable perhaps. Whether or not people adopt it, we'll have to see. The area that I'm much more interested in right now, though, is in therapeutics. And I think what's behind the whole precision medicine initiative, it's certainly what President Obama proposed, his million-person All of Us project, is that, you know, medical usage, medicines right now are killing us in a couple of ways. The cost of the health systems are too high, and they're making us sick a lot of the time. So if we can target the right medications to the right people, we'll be ahead of the game. And so if you take something like statins and blood pressure medications or anti-TNF for inflammatory disease, you know, not everybody's benefiting from those. Okay, so what's called the number needed to treat is really high. The number of people taking a drug to benefit just a few is really enormous. And if we can use genomics to say who's at risk or who's going to respond or who's going to have an adverse response, which I think are totally achievable in the next 5 or 10 years for most chronic diseases, then we can really reduce those expenses for the system, and we can really make sure that the medications are benefiting those who need them the most. And I personally, I think that's the way that genomics is really going to take us in the next 5 years.
So we have seen that there are lots of things that genomics still do not understand, but meanwhile we have lots of possibilities of using our knowledge.